Okay, so we're looking at the final piece of the protein story, looking at specialized proteins. So we've done all this stuff on structure. Let's actually look at how that structure links into function. So I have picked 4 proteins to work through, but obviously this can be taken broader and there are other proteins in your course. But we're just going to use these to highlight some of the examples of what we've been talking about over the last few weeks. So let's start off with enzymes. So when we talk about enzymes, we're talking about one of our classical proteins. And when you study enzymes, this is right after you study proteins so that we can build it all together and start applying the things we've got. Okay, so first off, we talked about amino acids and bonding, and we built that into primary, secondary, tertiary, and quaternary structure. And that's where we can now start building directly into enzymes. So remember, we had primary, that leads into secondary, that leads into tertiary, and that can lead into quaternary. But it doesn't always, it depends on the protein. So the big thing here is that it is these last 2 levels of structure, tertiary if we're stopping there, quaternary if we're going there, that then builds us into active sites. So even if you haven't got to enzymes in your AS Bio course, you should be familiar with the fact that an enzyme is a protein and an enzyme has an active site. A lot of what we do at AS is actually repeat things that were studied at IG, but in a whole lot more detail. So when we talk about active site, we're talking about the structure of the active site, and we're talking about the function of the active site. So the structure of the active site is affected by all levels of protein structure. Because remember, it's that primary structure that first links the amino acids together, and is then those R groups of the amino acids that can affect our tertiary quaternary structure. It's the OH and the NH groups on those amino acids that affect our secondary structure. So everything is building in to get the precise shape of that active site. Okay, and we talk about specificity. Specificity. It doesn't want to spell right today. Specificity. This is of enzyme for substrate. And this all depends on the structure of the active site. So if we change the structure of the active site, then we change the specificity, we have a different enzyme. Okay, if we think about active site in terms of function, remember those R groups? Okay, they aren't just a random collection of molecules, they have an impact on our tertiary and quaternary structure, but they are also what forms bonds with our substrate. And we have, take a wild guess, we want a temporary bond, we have H bonds, hydrogen bonds. You write out hydrogen when you're writing in an exam. Okay, because it's these hydrogen bonds that then hold that substrate in the enzyme. It's not just a case of we go together, we go apart, and sort of it happens in the meantime. That's what you're learning at IG. At AS, we want more detail. So substrate enters active site, hydrogen bonds between the substrate and the R groups. Then we can catalyze a reaction. We can stress on that bond if we're doing a hydrolysis reaction, for example, and we can bring the 2 substrates close together and have them in the right thing to catalyze that removal of the water for a condensation reaction. Okay, so all of these things are part of, every time we talk about a glycosidic bond forming or breaking, we're talking about an enzyme control reaction. So the enzymes are really out there and everywhere. And active sites don't work without hydrogen bonds between R groups and substrates. So it's all tied together in one big happy mess. Right, so function of the active site and structure of the active site are both linked all the way back to primary, secondary, and tertiary and quaternary structure. So for primary, we need to take note of the R groups. Secondary structure is the beginning of the specific shape. And then tertiary and quaternary are the completion of the 3D shape. So even when it comes down to things like enzymes or globular proteins, we're talking about tertiary structure. We're talking about having those hydrophobics on the inside, hydrophilics on the outside. Okay, so I'm trying to watch the time to make sure I can get 4 into 30. But there is one other important thing that we need to mention here when we're talking about enzymes, and that is about denaturation. Okay, so when we're talking about denaturation, we're in particular talking about temperature and pH. So when it comes to temperature, we're talking about destabilization of bonds. And pH, we will be talking about alteration of charges. So if you think back to your primary, secondary, tertiary, quaternary structure, you've got your peptide bond, you've got your hydrogen bond, then we're going to add in ionic bonds, we're going to add in hydrophobic interactions, and disulfide. Ooh, it just fit. So what you've got to think is, of each of those bond types, what is destabilization going to affect the most? If we increase vibration within that molecule, which are the first ones that are going to go? Which are the last ones which are going to go? Same with pH. If we alter the charge environment of the enzyme, which of our bond types are sensitive to charges? So they're going to be the first ones to go. Okay, all right. So few highlights of enzymes and how we can link this into protein structure. Any burning questions about enzymes? Or can we move on? Okay, so there's no fast typing of burning questions. So we shall move on now to talk about antibodies. So antibodies only come up at the very end of your course. So why are we discussing them now? It's that awareness, it's that application. Okay, and the thing is that by the time you get to the end of the course, you're so excited about human biology and diseases and things like that that you forget, I did molecular stuff too. So it's important that you have that sort of constant theme of molecular coming through. So yes, we take it up to a bigger scale, but at the same time, we're keeping that molecular detail, that molecular understanding of where we started. So antibodies. Let's do this. Oh, goodness. This one goes like that. This one goes like that. Okay, so something like this, okay? And what we have at the top here is a binding site. I'm going to try and avoid using the super technical terms now because you learn a lot of technical terms in immunity, but a lot of you aren't there yet. So we'll keep it simple for now. Okay, so binding sites, there's 2 of them, and we have 2 polypeptides, 2 different types. Okay, so we're also talking proteins. So if we're talking proteins, we're talking binding sites. Here is our direct link into enzymes. We have, again, specific shape. Again, we have that primary, secondary, tertiary, and here we go. We have two different types of polypeptides. We have two short ones and two long ones. Multiple polypeptides, we most definitely have quaternary structure when we are talking about antibodies. Okay, so here again is one of your nice examples for bringing it back. Okay, so when we talk about antibodies, we're talking about specific shape of the binding site, which then affects the function of the antibody. Okay, they're also temperature sensitive, pH sensitive, same like enzymes are. And the other thing that it's nice to pull in here is about diet. Okay, so what we're going here is all the way back to sources of amino acids. Okay, so if we think about diet, we need a protein source. Well, more than one. We need protein sources, different proteins. This gives us amino acids. Okay, because remember, we can't synthesize amino acids. They're built and then we have to consume them. We have to break down our protein, get our amino acids. So there we go. We're revising hydrolysis of peptide bonds. Protein consumed. Okay, then we're going to transport the amino acids. And then we are going to build antibodies. So let's link this to organelles. Our cells that produce a lot of antibodies, so particularly in plasma cells. Okay, this is our cells that build our antibodies. And what they have is a huge amount of rough endoplasmic reticulum. So remember, we talked about the fact that rough endoplasmic reticulum is going to be responsible for building those proteins that are going to be exported. So in ribosomes on the outside, you get your polypeptides synthesized with the peptide bonds, and it goes into the RER, and then it's modified and all of that stuff that we talked about when we talked about organelles. So here is our prime example. We have to have lots of RER. So when you look at plasma cells and their organelles, and you often find you got a cell, and then it is just packed with, I'm not going to draw it in detail, but it looks something like this. The cell is, yeah, it has a nucleus and other organelles as well, but it's the cytoplasm, you don't see blank space. Okay? You see rough ER all over the place because it's building so many antibodies. Okay, so we have the specific shape, we have binding sites with specific, we have variety because we need different types of antibodies. So all we have to do is change that polypeptide. We change the sequence of amino acids, change that primary structure, and we give rise to different types of antibodies. Okay, so again, you're linking from the very beginning all the way to the very end. Any burning questions on antibodies? Or shall we move on to membrane proteins? Okay, right, no questions have popped up yet in that split second you had. So let us move on to talking about protein structure in regards to membranes. Okay, so if you think of your cell membrane in your fluid mosaic model and all the hundreds of phospholipids that are a pain in the neck to draw, so we're going to draw very beautiful phospholipids. Of course, you would take a little bit more care if you were doing this for an exam answer. If you haven't got to the fluid mosaic model, never fear, it is coming. And then you will understand what these particularly awful scribbles are actually representing. So very briefly, we have a phospholipid bilayer. And then I've just given 3 different types of proteins. Okay, so here we have what is called a channel protein. And here we have a carrier protein. Let's take this outside and this inside. And then let's make this one with a radius, with an aerial even, which makes him a glycoprotein. Okay, so a few things to sort of highlight when we're thinking about cell membrane proteins with regards to protein structure. So we're not looking at this in terms of transport across a membrane, because our focus on this little mini series has been on proteins. So channel proteins, carrier proteins, we've drawn them different shapes, and again, it's specific. We have specific shapes. Okay, so think back to your IG, transport across membranes, when the life and everything was simple, and you just had active transport, and that involved the carrier protein. End of story. So life is going to get much more complicated. We're going to have diffusion and facilitated diffusion and active transport and changing formation, and how and why does a protein change formation. So we're going to touch on that one for a moment. In some cases, our carrier protein will be for active transport. And in other cases, he's going to be for facilitated diffusion. So casting back your memories, active transport requires ATP, and it's the ATP which makes the protein change shape. Okay, facilitated diffusion, no ATP. Right, so what happens here is that the molecule that we're going to transport binds to the protein. Now, think about enzymes and think about inhibition. So when you do enzyme inhibition, you find out that you can have an inhibitor that binds somewhere else on the enzyme. That then changes the shape, because remember, everything is bond dependent. So if we go and stick something else in, it's going to change somewhere else on that protein molecule. So in the same way that we can stick an inhibitor on and change the shape of our active site, if we stick a molecule onto a carrier protein, we can change the shape of the carrier protein. And that means then our thing that's being transported can be flipped over onto the other side of the membrane. So this causes change in protein conformation, we call it. So we want to use those fancy words, okay? So it changes the conformation of the protein, and then we can release on the other side of the membrane. Remember, we would have different proteins going in different directions here because of the specific process. Okay, so that's one nice thing when we think about the specificity of those bonds within our protein. If we alter the bonds at any part of that molecule, it can have knock-on effects. We can also effectively denature these proteins if we have them at high temperatures or at high pH. It's a beautiful experiment with beetroot. And what you do is you stuff up all the proteins in the membranes, and then of course all the pigment from inside the beetroot leaks out, and then you get very pretty, beautiful, different colors of test tubes, different shades of pink, depending on how much you've stuffed up the proteins in the membrane. Okay, so it's all linked together. It's all that same structure of that protein. It's all coming back to primary, secondary, tertiary, quaternary. So the other thing we're touching on here, of course, is our glycoprotein. So here we're going back to how did we make a glycoprotein? Okay, so remember, we went ribosome to RER, and we stuck on carbohydrate chain. Then we went to the Golgi, and we modified that carbohydrate chain so that it's individual and it's specific. And if you think about it, it works really well if we do this, because now we've got this protein and we've got these phospholipids, and we can put things into our cell membrane. So it all travels through very beautifully. And again, all of this gives us specific shape. And when you have specific shape, you have specific function. And these guys are often involved in things like cell signaling, so we've got that idea that we have a specific shape, so we can respond to a specific signal, and you get into the full details of a bit more of how that actually happens when you get into looking at transport and communication across the cell membrane. Okay, so there is a lot more to learn about what we're doing here. Another reminder, we're highlighting things that link into protein structure and the importance of understanding peptide bonds and primary structure, secondary, tertiary, quaternary, building it up. How does this all link together? So there's one other thing I want to really touch on here, and that is linked to the structure actually of our phospholipids. So our phospholipids, you can see we've got circles on the top and the bottom and squiggles in the middle. So the circles are representing our phosphate groups, the heads of our phospholipids, and those are hydrophilic. They like being against water. So they're happy against the cytoplasm, they're happy against the extracellular fluid. Okay? What those squiggles are representing in the middle is our hydrophobic lipid tails. So remember, phospholipid, phosphates and lipids go together. So the core of our membrane is hydrophobic, and that is actually a large part of how we control what goes in and out. What it means is that for any protein, any part of the protein that is in contact with the central core of the membrane needs to be hydrophobic as well. Otherwise, it's not going to sit there. It's just going to bump out. Okay, so any piece of the protein that is in contact within that core of the bilayer must be hydrophobic. Okay, so why do we draw it like I'm scribbling now, that it's sort of just the outside? Well, if you think about it, our channel protein, right? We've got things coming through which are not necessarily hydrophobic. It can be a hydrophilic pore. That could be a channel protein, which is a for water, an aquaporin. Okay, so specially designed to increase the diffusion of water. So we can't have a hydrophobic core in there. We've got to have a hydrophilic core. So again, okay, it's not just when we're talking about protein structure, it's not always that we have hydrophobic groups on the inside, hydrophilic groups on the outside. Yes, often when we're talking about enzymes, because you think enzymes are in cytoplasm, they're in a wet environment, they need to be hydrophilic on the outside. But when it comes to channel proteins, glycoproteins, carrier proteins, those membrane proteins, then hydrophobic regions have to be on the outside, where we're in contact with the phospholipids. So, hydrophobic regions. All right, so we have 4 minutes left to tackle hemoglobin, which gives us a new, sort of another set of things. So, burning questions on membrane proteins, or are we all cool? Okay, right. Silence is golden. Let us move on for the last few minutes to hemoglobin. So, now you might be breathing a sigh of relief because you've done proteins and you've already touched on hemoglobin and you know where this is going. Okay, so we have really drawn from throughout the course when we talked about these proteins today. Hemoglobin is a classic globular protein. And he's also our classic quaternary structure. So, why does he have to be globular? Let's think about that a little bit more. Okay, he's got hydrophobic inside, hydrophobic outside. Why? Well, where do we find hemoglobin? Hemoglobin is in our red blood cells. Take it further. Where is it? It's in the cytoplasm. NK, and your cytoplasm is an aqueous environment. Oopsie. As soon as you have an aqueous environment, then you need something which has hydrophilic on the outside. Cells are going off, sickle cell anemia. One of the problems with sickle cell is that our hemoglobin swaps a hydrophilic for a hydrophobic amino acid, primary structure, all the way back down, okay, to the very, very beginning. And very beginning, instructions in DNA, mutation of a base pair, changing then all our instructions for building this protein, changing ultimately the shape of the protein. So a little aside here, taking this to our transcriptions and translation discussion, keeping it simple for those of you who haven't got there yet. Doesn't seem like my spelling is doing too well when I'm sprinting. Okay, right, so we've linked now to transcription and translation. We've linked to errors in our code, errors in our instructions, all the way up to why do we need a hydrophilic on the outside protein. So classic quaternary structure, we have 2 alpha polypeptides. Okay, let's be well behaved. 2 alpha globin polypeptides. And we have 2 beta globin polypeptides. And we have 4 heme groups, which is our Fe2+, okay, enabling us to carry the oxygen. So this is the thing where we've got a classic quaternary structure. We have multiple polypeptides, different polypeptides, and prosthetic groups, which is why we like hemoglobin for our classic quaternary structure. Okay, it is now 10:01, so I'm going to have to draw a line, but this should hopefully give you an idea of how much there is. Even if we just think about proteins, it's not just a case of primary structure is. Okay, learn it in context and start building those links with the other topics, just to solidify everything. Okay, so if you want the scribbles from the session, please make sure you send me a message, because otherwise I will get distracted and I will forget. I'm not making promises to remember. Otherwise, have a wonderful rest of the day. I'm grabbing tea and then I'll see you for maths if you're coming for that. Have a great week.